So, welcome back to the second part of the TIPWG 101 webinar. And this section, we are going to be talking about the TIPWG funded research. And so in a minute, I am going to hand you over to our host, Jolanda. So Jolanda, if you come online, turn on your camera, unmute your mic. There is a slight change to the schedule because we have Professor Talbot's student, Jina Park, giving the presentation on his behalf, which is quite exciting because I'm not sure how many of you will know this, but on Sunday it is the International Day of Women and Girls in Science. And it's all about promoting, but also facilitating and trying to elevate this as a career for women and girls. Because it's still an area where gender inequality is a huge issue globally. So to have a section of research brought to us by a female researcher with 3 presentations done by female researchers a few days before this International Day is quite a good thing. And it's quite exciting, especially considering we're often thought of as a sector dominated by men, which is obviously something that we are trying to address going forward. So Jolanda, I'm going to hand it over to you to say a bit about research, and I really look forward to listening to all 3 presentations.
Thank you very much, Katie. Yeah, good afternoon. Well, nearly afternoon, everybody. I think we've got quite an exciting lineup of presentations in this session. But before we get into those, just a little bit of a background to the TIPWG research component. This is really research that's aimed at being more technical type of research, so bridging some of the academic results and knowledge with the practical implementation of especially pesticides in the industry. So this research that's funded by TIPWG via FSA mostly, it really deals with pesticides and how we can use it effectively in our nurseries and plantations. And it's aimed to span research across all company needs. So in light of that also, because TIPWG is a community network or working group also, we encourage you to communicate with us if there are any specific technical type of research questions that you and your company think we should address. Then we can have the discussions and look into the way forward in terms of getting some of these research projects realized. So some of our past projects included, for example, looking at the environmental fate of pesticides in water and soil. And you'll get an update on this from soon-to-be Dr. Ndlovu. I'm pretty sure that's going to be the outcome of her Ph.D. that she submitted in December. And we'll leave some questions and so on to her after her presentation shortly. Another very successful past project was the development of economic and environmental and social pesticide risk models for plantations in South Africa, which was driven by Dr. Jonathan Roberts and Professor Keith Little. But for today, we will in this session focus on 3 research projects. A new one, which was established last year through Forestry South Africa funding, includes specifically a project that aims at trying to increase our suite of fungicides and insecticides that we can use in our nurseries. Now, sometimes silvicultural and hygiene interventions just aren't enough for us, and we need to revert temporarily to pesticide use. So our problem with this, as many of you are all too painfully aware, is that there's very few products that's actually registered for use in plantation forestry, and especially in our nurseries. And this really exposes us potentially to resistance buildup and other issues. And also, some of these products, the few products that we have registered are quite old, and there might be new chemistries and better products out there. So TIPWG is partnering with especially chemical companies, and with this I include biological companies as well as the companies producing the synthetics, to look at other products that's out there and already registered, especially in agriculture, and which we could potentially assist in label extensions for inclusion in our forestry suite of products. So we'll be hearing from, first of all, from Noxolo Ndlovu, which will tell us more about the soils and water fate of pesticides in plantations. And as I said, this is a project that she did for her PhD, which she submitted in December. Currently, Noxolo works at NCT as a research scientist, looking especially at tree improvement. The second project we will hear about is one that started last year and which is run through the ICFR. And the project aims to expand the suite of insecticides and fungicides that we can use in our nurseries. This project will be presented by Dr. Bernice Bancole. She's a postdoctoral research fellow at the ICFR. And she holds a PhD from UKZN, and she has extensive experience in biological control of agricultural diseases and pests. So we're very excited to have her on board with the TIPWG and the Forestry South Africa funded project. The third project group is managed by Professor Bruce Talbot at Stellenbosch University. And there are 3 projects related to pesticides that he's managing. This includes one that looks at the feasibility of various ground and aerial techniques for pesticide applications in plantation forestry. And then also a project to develop and apply machine learning or AI methods in digital imagery using drones and satellites. The project we will hear about today is that of MSc student Jina Park, who's looking at applying machine learning techniques in identifying weeds and vegetation types with drone imagery. Jina has a BSc in forestry, and she's currently busy with her master's on this project. Before I hand over to the first presenter, just like to encourage everybody and remind everybody that you can post questions to the presenters in the question and answer box. So please remember to do that so that we can have some feedback to our presenters also. So with that, I'm going to hand over to our first presenter so that you can hear the interesting stuff and stop listening to me.
Thank you, Jolanda. Sharing my screen. Okay, I'm sure you can see that, right? Jackie, Aiden, it's all visible?
Yeah.
All righties. Okay, thank you so much. Thank you, Jolanda, for the introduction, and a good day to everybody present. Congratulations to the TIPWG team for organizing such a successful webinar, and I'm really grateful for the opportunity to present on such a platform. So as already mentioned, the work that I will present today was completed as part of my PhD project through the Nelson Mandela University under the supervision of Professor Keith Little and Drs. Brenda Bailey and Carol Rolando. The project was entitled Towards Understanding the Soil and Water Fate of Pesticides Used Operationally Within South African Plantations. It's a quick presentation outline. Initially, I'll just give a brief background of the study, and then we'll spend the bulk of our time looking at the research objectives, looking at the data collection, the results that are coming out from the work, and then I'll end off with the overall conclusions. So pesticides are considered as a last resort or last option when managing destructive pests and diseases within forestry plantations. Pesticides are an important component within integrated pest and disease management programs, and they are likely to remain important even in the future. But the plantation forestry industry is restricted in terms of the number of active ingredients that are approved for use, particularly forestry plantations that subscribe to forest certification standards. And this is a concern because more often than not, the forest certification schemes approve or disapprove the use of different pesticides based on conclusions from laboratory-based studies or field studies that have been conducted in environments that are not reflective of the South African forestry landscape or operational practices. And both of these are not ideal because the lab studies are likely to overestimate the risk, whereas the field studies conducted elsewhere might misrepresent the risk that would occur under South African forestry plantations. In general, there are a limited number of studies that have addressed the environmental fate of pesticides within forestry plantations, especially forestry plantations in the Southern Hemisphere. And so the purpose of this project was to investigate the environmental fate and behavior of pesticides used within South African forestry plantations, taking into consideration the manner in which we use the pesticides and under our South African environmental conditions. In the context of this research, investigating the environmental fate and behavior meant that we sought to understand the following. Number one, at what concentrations do the pesticides occur in soil, water, and sediment following application? Number two, how long do those pesticides remain in the soil, water, and sediment following use? With that, we really wanted to understand whether the pesticides are degrading rapidly post-application or they are sticking around in the environment for extended periods of time. And then thirdly, in the soil, we wanted to get an idea of whether the pesticides were moving down the profile, again, just to sort of gauge whether the pesticides that we apply are susceptible to leaching. So understanding the environmental fate and risk of pesticides is important because data from such studies can be used to inform responsible and sustainable forestry management. This study had 4 research objectives. Objective 1 was completed as a literature review, which evaluated the environmental behavior, fate, and risk of key pesticides used within South African forestry plantations. This literature review confirmed that there were no published environmental fate studies that have been conducted within South African forestry plantations, and so it sort of supported the need for the work I will present today. This literature review was published in the Southern Forest Journal in 2022. The second objective investigated the soil fate of key pesticides used within South African forestry plantations and the risk posed by their use to the soil environment. The third investigated the water and sediment fate of pesticides and the risk posed by their use to drinking water quality, non-target water and sediment dwelling organisms. With the fourth objective, we developed a decision support tool that the industry can use in conjunction with the ISRAs to guide pesticide use decisions. To be honest, this objective requires a presentation of its own because there's just a lot to explain. But for today, I'll just give a very brief overview of everything that is going on in that objective. I'll be very brief. Research objectives 2 and 3 were based on a field study that we implemented at NCT's Ingwe plantation in the KwaZulu-Natal Midlands. The site was 16.6 hectares and it was an ex-eucalyptus site replanted to Eucalyptus smithii in January 2020. The site was considered representative of typical forestry sites in a sense that it was representative in terms of climate, of soils, as well as the species planted. In addition to the site being representative, it was selected because it could represent a worst-case scenario for environmental contamination because the site was near a small stream. The site was steep with water-repellent soils, and so facilitating that increased risk of runoff from the site to the stream. Also, the slash on the site was burnt, meaning there was little organic matter that could absorb the pesticides and so preventing them from leaching or running off. And then also, as already mentioned, the site was reestablished on a previous eucalyptus stand, which then meant that more chemical would need to be applied in order to kill the stumps of the previous crop. 4 plots were strategically established on the site in which, following any pesticide application event, soil samples were collected at predetermined intervals. The soil samples were collected at the 0 to 10 centimeter depth and at the 10 to 50 centimeter depth. Remember, we wanted to answer the questions, at what concentrations do the pesticides occur in the soil post-application? We wanted to determine how long the pesticides were remaining in the environment, and we also wanted to determine if there was a risk of leaching. In addition to the 4 plots for soil sampling, 3 water sampling points were also established along the stream. S1 there, I'm not sure if our cursor works. Okay, the cursor doesn't work and we don't have a pointer, but S1, oh, does it? There we go. Thank you, Roger. Okay, so it does work. Okay, S1 there was above the study site, and it represented a portion of the stream that was uncontaminated by pesticides. S2 was just below the study site, and then S3 was further down. C1 to C4 were drainage culverts where water runoff flowing directly from the site was collected before the water could enter the stream. Water and sediment samples were collected following the first 2 rainfall events of 10 mils or more post-pesticide application. This study was carried out over a period of 26 months, therefore covering the period from pre-plant spray to canopy closure, which within South African forestry plantations is the period of the most intensive pesticide application. Over the 26-month period, there were 7 pesticide application events, and this included 4 herbicide events, 2 insecticide events, and 1 fungicide application event. Although no pests and or pathogens were observed on the site, but just to fulfill the study objectives, we applied the fungicides and the pesticides anyway. Before I get into the environmental fate results, I'd just like to quickly highlight that glyphosate was the most applied pesticide at Ingwe, not only in terms of like the number of applications, but also the quantities applied. Over the study period, a total of 124 soil samples, 43 water samples, and 23 sediment samples were collected. Now onto the environmental fate results. As I've shared in previous presentations, the most exciting results, and I guess the most important result that has come up from this work is the fact that glyphosate was never detected in any of the soil samples collected. And this is despite the fact that it was the most frequently applied and at the highest quantities. But to be honest, this result observed at Ingwe is not surprising at all, because numerous publications, they do report the rapid degradation of cypermethrin, of glyphosate in the field. I guess the moist and warm weather conditions that prevail during pesticide application seasons, as well as the mild temperatures observed in the winter, possibly facilitate the rapid degradation of glyphosate in the field. As for the other pesticides, they all showed very similar trends. The results show similar trends. Firstly, pesticides were consistently lower in the 10 to 50 centimeter depth when compared to 0 to 10 centimeter, that is the topsoil horizon. Maybe that might be indicative of the fact that the active ingredients that we use are not susceptible of leaching. And again, that is in agreement with what literature has been noting or reporting regarding the active ingredients that were investigated at Ingwe. Secondly, the pesticide ingredients, they showed a decline over time, and they had reached below detection limits by the time of the final sampling. With regards to water and sediment, what we found is that across the different pesticide application events, the highest concentrations of pesticides reported in water were always or occurred right after the first event, rainfall event, post pesticide application. At the next sampling event, pesticides had decreased or were below the detection limit. What we also found is that cypermethrin was never detected in any of the water samples collected. And this was, again, like 100% expected, because cypermethrin has a very high absorption coefficient. And so in any water and sediment system, cypermethrin will quickly adsorb to the sediment and therefore are leading to reduced concentrations in water. Samples taken to quantify the quantities of hexazostrobin post-application were unfortunately compromised. And so at present, we don't really have an idea. We don't have any information regarding the hexazostrobin concentrations in water following its application. With regards to the sediment, what we found is that with the exception of glyphosate, which was never detected in any of the samples, all other pesticides were detected. And in sediment, we found that they persisted much longer in sediment than they did in water, which was again 100% expected because literature is reporting longer persistence of pesticides in sediment when compared to water. But these pesticides in sediment, they did show a gradual decrease over time, and they had reached detection limits, below detection limit by the time of the final sampling. So now that we have an idea of the environmental fate of pesticides at Ingwe, the next question to ask ourselves is, were the pesticide concentrations recorded toxic? And to help us answer that question, we compared the concentrations reported at Ingwe to lethal concentration values derived for standard aquatic and soil organism test species. And these lethal concentrations are normally derived from laboratory studies. And so in a way, it makes them like the worst case values to use. For soil, what we found is that the pesticide concentrations at Ingwe, for the most part, posed a low risk to non-target soil organisms. This is because the concentrations were well below those lethal concentrations reported to cause adverse impacts in the soil environment. But we were very conservative with hexazostrobin in that we concluded that it poses a moderate risk because the hexazostrobin concentrations at Ingwe of that 0.05 milligram per kg were very close to this 0.1 milligram per kg that the laboratory study showed that it reduces the biomass of fungi and bacteria. But to be honest, when you consider the fact that these impacts reported in this laboratory study were short-lived, and that hexazostrobin in the field is likely to be adsorbed to mineral and organic surfaces, I could even say that the risk that hexazostrobin poses in the field is also very low. With regards to the water, the glyphosate and triclopyr concentrations, they posed a low risk. Hexazostrobin, of course, the risk is unknown because we couldn't quantify the concentrations of hexazostrobin following application. Unfortunately, though, metazoclor posed a high risk at Ingwe because the concentrations were like sky high. They were way above the water drinking standard, as well as the lethal concentration for the most sensitive aquatic species. Cypermethrin, I put a question mark there because evaluating the risk of cypermethrin based on water concentrations is not really the most reliable. Cypermethrin risk is a bit tricky. Yes, it does. I mean, cypermethrin does adsorb to sediment very quickly, and so it becomes biologically unavailable in the water column in a sense. But for those short durations that cypermethrin is in the water column, even at the lowest quantities, it can still cause devastating impacts. A lot of field studies do support my statement right now. And so with cypermethrin, I think the risk is just a bit tricky. Using water to conclude on the risk that it poses is actually not the best. With sediment, again, glyphosate and hexazostrobin, glyphosate and metazoclor, they pose a low risk. Hexazostrobin and triclopyr, the risk is unknown because there were just no studies that linked sediment-derived triclopyr and hexazostrobin concentrations to ecotoxicity effects of sediment-dwelling organisms. So there's a gap in the literature there. Cypermethrin, unfortunately, also just again here, it poses a high risk to sediment-dwelling organisms. So cypermethrin is a bit of a red flag currently. That brings us to the conclusions of the field study. For the most part, honestly, these results are encouraging, more especially because they do indicate that glyphosate, which is the most used pesticide within South African forestry plantations, poses a low risk. The results also indicate that except for metazoclor and cypermethrin, which pose a risk, but all the other pesticides, when used according to label recommendations, they pose a low risk to drinking water quality and non-target soil and aquatic organisms. Ingwe was a worst case site, and so we might expect that metaxachlor and sapamethrin impacts on those sites will be, on other sites might just be lower. From the field study, we can conclude that pesticides can be safely used as a part of IPM, but as we use these results, we do sort of need to be conservative. We need to acknowledge that these are from a single study in one region and over one growing season. Almost want to just say thank you, hand back over to Jolanda and just leave it here, but I cannot, because in addition to the field studies, we also developed a generic decision support tool that the industry can use. As mentioned at the beginning, this just requires a presentation of its own because it's just quite a bit to explain, and so I'll just keep it brief as much as possible. So there are a number of pesticide risk assessments that are currently available, but what we found is that these pesticide risk assessments are mostly very broad, very general, very complicated at times and data intensive. And so what we wanted to do was to create a tool that was simple and practical. But most importantly, we wanted to create a tool that only bases the evaluation of risk, of pesticide risk, on field observations, not just taking laboratory studies, but only looking at what has been observed in the field. Secondly, we wanted to create a tool that was tailored to the South African forestry pesticide use. The generic decision support tool that we created is relevant to SA forestry, and it's also realistic because it bases the risk evaluations on field reports. The building blocks of the decision support tool. So as a foundation, we considered the inherent toxicity of pesticides, which is mostly derived from laboratory studies. And this was acceptable because the inherent toxicity of pesticides does give us an indication of the likelihood of the pesticide to cause harm. Secondly, we considered degradation values. So just considering how long is the pesticide expected to persist in the environment. And then thirdly, we considered field-based reports of pesticide risk. So within those, we were looking at has there been any adverse impacts that have been reported under field situations following the use of particular pesticides, and how long do those adverse impacts last? And is there recovery post the use of those pesticides? So we're sort of negotiating such questions and looking at field reports that have reported actual impacts. And then following which after looking at the field reports, so in light of those field observations, we then ask the question, how does the South African forestry specific use patterns influence that risk? And so in that, we considered the methods of application as well as the frequency of application in South African forestry plantations. Without going into any detail, what you see on the screen is an example of the decision support of the DSS that we created that was evaluating the risk that is posed by sapamethrin use. So we asked some questions, look at the literature to respond to those questions, and eventually we reach a conclusion of just the best actions to take with regards to the risk that any pesticide poses. Together with the DSS, we also put together two tables that provide details of the key drivers of risk, of pesticide risk that are meant to enlighten the foresters, especially on the ground, as they make decisions around pesticide use. Within the tables, we also included some recommendations of risk mitigation that the foresters can consider as they make those decisions for environmentally and I think responsible pesticide use. That brings us to the end of my presentation. So honestly, the work that I've presented today has provided a valuable first step towards understanding the environmental fate and risk that pesticide use poses within South African forestry plantations. I believe that it has answered some key questions that the industry has had for some time, but there are still some gaps that need to be addressed in order to provide a more complete understanding of the environmental threat and risk that South African pesticide use poses. For one, additional studies with more comprehensive sampling can be implemented across major site types and silvicultural regimes in South Africa. Secondly, studies exploring the risk posed by pesticide mixtures and degradation products can also be implemented, together with studies that derive degradation values relevant for South African forestry and models that can accurately predict the pesticide environmental fate and risk. Those should also be considered. And there's still a major research gap in the field of sediment toxicity. So future studies can also look into that. And as a final recommendation, we recommend that research into non-chemical pest and disease management should continue to take precedence, because pesticides are chemical compounds and they do have the ability or the potential to negatively impact on the homeostasis of the natural environment. Before I hand back to you, Jolanda, thank you so much, everybody, for your attention. And I would really like to just extend a massive, massive thank you to the sponsors and the supporters of this work, NCT and Nelson Mandela University, FPM-CETA for sponsoring my studies, TIPWG through Forestry South Africa for all the millions spent on the pesticide analyses, my supervisors, Roger Poole, the ICFR, and every single person that played a key role in making this work a success. Thank you so much, and I hand back to you.
Thank you very much, Noxolo, for a very well-presented talk. Amazing results. Very relieved for us as a forestry industry with the results that you've presented. An amazing amount of work that you managed to get done. And I think just from my side also, and the same for the following presentations, is just a big word of thank you also to the pesticide companies that have contributed to some of these studies. So Noxolo, I don't see any questions in the chat box or in the question and answer box, but I do have one for you. Maybe I missed it, but here we go. Just want to ask specifically also, your decision support system, where is this available? Is it available already for forestry companies and other users to see, or is that still going to happen? And in which format is it available to people?
All right, thank you, Jolanda. Actually, it's a very good question. I forgot to mention it in my presentation. The plan was to mention it, but and we submitted the chapter that addresses that objective to Water SA Journal at the end of last year. So we're just waiting for acceptance and publication, and as soon as that's available in the public domain, I'll also just hand it to TIPWG, and maybe it could be available on the TIPWG and TIP website. Yeah.
Thank you very much, Noxolo. I'm sure everybody's going to jump at using it, because it's incredible work and important for us. Maybe a follow-up question. And you tested a certain number of products in your study. You did mention that it would be ideal to do your study on different sites, because obviously you did it in a study with a specific soil type and environmental conditions. But also, do you think this type of study needs to be done for all the active ingredient types that we use in plantation, or would it be representative enough for us?
I think, of course, the ideal would be that it is like each active ingredient is tested, because the fate of each active ingredient is not the same. Each active ingredient has got its own physical chemical properties, it's got its own degradation values and its own toxicity. And so most definitely it would be ideal to test this out with all the active ingredients that we are using. But at present, fortunately, we've got a very few active ingredients that are approved. And I think as the industry, it would be affordable. It would be affordable.
Jolanda, can I just add one thing quickly? The products that were chosen at the time were the ones that were most used in forestry, being the glyphosate and the cypermethrin. We then chose metazoclor because pre-emergence were starting to be used, but we didn't know which ones and how many. And then azoxystrobin being the fungicide that was used, we put those in just to test and have a look-see. So that's kind of like how we made the decision on that. But yeah, we need to expand the project onto a much larger scale than what we did at Ingwe, and then look again, do a survey to see what are the most popular, most widely used products happening, and then start doing those kind of tests, bring those products in along with the ones we did test to see if we get different results.
Thank you, Roger. Thank you, Noxolo. Yeah, I mean, there's always so much more work that can and should be done, and the toss-up between people who can do the work competently, like you did, Noxolo, and the funding to do this type of work. So thank you to you and to Roger and the team for pushing this type of work, and we look forward to what we as an industry can do going forward based on your recommendations. There was one question in the chat box from Josiah. Thank you for that, Josias. But I think you've answered his question, which dealt with, are you going to look at replicating the study in different regions and provinces? So Josias, I think the answer there is yes. We need to look at how we're going to make that happen. Then there's a question from Richard Ferguson. You indicated that field-based reports of pesticide risk helps provide data. Is that collected by industry, and how much data is there, and was this focused sampling? Should I repeat it?
Yes, please.
Okay, so the question was, you indicated that field-based reports of pesticide risk helps provide data. Now, is that data collected by industry, and how much data is there, and was this focused sampling?
Okay, so the first part, Richard, there's not a lot of studies out there that have been conducted in the field, and hence the need for the work that we carried out at Ingwe. Our major concern, I think I speak from the industry's perspective, is that when the forest certification schemes approve or disapprove the use of different pesticides, the decisions are never based on what has been observed in the field, but it's always from laboratory-based studies or studies that have been carried out elsewhere in North America. And that is not really reflective of the risk that could be observed within South African forestry plantations. And so that restriction of that particular pesticide is really unfounded. Then I think going into the future, it's quite broad. Even the forest certification bodies, they need to change their criteria, how they arrive at the conclusion of restricting or approving the different pesticides. So from their side, before they can get to the conclusion of approving or disapproving, where does that data come from? Why do they say that it needs to be disapproved or approved within South Africa? So I guess it would be definitely an effort from their side, but us as an industry too, because we are proactive, we are wanting to manage responsibly, we are wanting to make sure that our practices are sustainable. So even for us as an industry, it would be, I think, a collective effort really to make sure that we are using field-based data to make more informed decisions. And I think the last one is how much data is there, and was this focused sampling? Within South Africa, this was the first study that I'm aware of that was carried out, the first study of this nature to understand the environmental behavior of pesticides in the soil. Maybe different companies have done work that is maybe not published, that is private, but just as an effort from the South African industry, this was the first work of its kind. So there's really not a lot of data that is out there. Yeah, I think I am answering that.
Can I just add there, just for Richard's benefit, because he's new to the whole FSC thing, all of our pesticides, when we get them in South Africa, their half-lives have all been based on studies used where the product has been manufactured, and the majority of those are in the Northern Hemisphere. So we've got a difference already between the North and the Southern Hemispheres. And then we go into, if you look in the Northern, some of their soils can actually be frozen for quite a few months of the year. So the whole breakdown process really gets slowed down. Whereas you move to South Africa, yes, we do get quite cold in winter in certain places, but the majority of our country, there is still activity happening in the soil. So for us to understand products that FSC are putting on the list and saying we can't use them because of this half-life and this toxicity. We hadn't had data to actually look at this kind of stuff. And this is where the birth of the whole project came from, is for us to get a better idea of what we are doing. And we took the example that was done in New Zealand with Brenda and Carol, where they actually ended up having some products after doing the testing put back onto their list that they could actually use. So this is where the birth happened, Richard, and we're trying to get data that is substantial so we can actually use it, because we don't really have a nice library available of this kind of data in South Africa.
Thank you, Roger Noxolo. I know we are taking a lot of time, but I think it's such important work. So I'm going to deal quickly with, there's a question and a comment from Richie Morris, which deals with the scientific integrity of the sampling process, which he says may need some consideration so that similar procedures are followed that stand up to industry protocol. And then he has a follow-up on that, is which standards are used for hazard and risk assessment, example in domestic water use. Is it WHO or SABS?
Which standards did we use?
It was WHO.
WHO.
And also the pesticide properties database has published a lot of information regarding the lethal concentrations of pesticides. And that was the main basis of comparison and just approaching literature values as well.
Thanks, Noxolo. I think so. With time in mind, I think you can also look at the question and answer box and maybe answer in more detail there, or I'm sure people are welcome to also contact Noxolo or Roger, one of the supervisors that assisted Noxolo and worked with her to get more detail. So Noxolo, once again, thank you very much, and we look forward to using your title soon. Good luck with your future work. So I'm going to hand over without further ado to Dr. Bernice Bancole from ICFR to present on her work. Thank you, Bernice.
And I think Aidan is going to share the screen. Thanks, Aidan.
Okay, thank you, everyone. Good afternoon. And thanks to the organizer and for Jolanda to do the great introduction about each of us. So through my presentation, I will give firstly a brief introduction and background. I will give the aim and the objective of the project and the progress we have made so far, the outcome, and a summary of the work plan we have for this year. So I'm Bernice Bancole, like Jolanda said, and I will provide you through this presentation an insight into the SIF project, which is related to the testing of alternative chemical and biological control products for the use in an integrated management of disease and insect that affect forestry industry. So the project has been implemented since last year, March 2023, and is a 4-year project funded by FSC through the TIPWG. And it is strongly relying on the collaboration between research institutes and the forestry industry partner. Is it the next slide? So like I said, we have the collaboration within research institute and forestry industry partner. Next. So from what we have learned, managing forestry plantation to maintain them in healthy, productive condition is a major challenge to forester and an integral component of sustainable forest management. And the use of pesticide is one of the available management tools where the disease and the pest can be controlled or excluded. However, there has been a limited pesticide toolkit for an effective control of those pests and disease. And on top of that, recently, there is a restriction to the use of the registered chemical product due to the social and environmental concern, together with the certificate body that the South African forestry industry subscribes to, and a delay in pesticide registration. So all this has unfortunately resulted in the availability of the less effective product, together with an associated decrease in pests and disease related to the productivity. So therefore, there is an urgent call for an alternative product that can be incorporated in an integrated pest and pathogen program. So the aim and the objective of this project, please, Eden. Next slide. So the aim of this project is to identify new pest management products for the use in the forestry industry, I mean nursery and plantation. Next, please. So specifically, the specific objective of the project is to collect or review the literature that is relating to the use of the product for the control of pests and disease. Secondly, to develop a standardized protocol for the nursery and the field testing of various alternative products. And lastly, to conduct nursery and field-based testing of alternative products for the control of the prioritized disease and insect. Next, please. So what have we done so far? So to achieve those specific aims, we firstly start with the survey and to provide you an accurate update of information related to the pests that occur in the nursery and plantation, which poses a threat to the forestry industry, like I mentioned earlier, together with their management and strategy. A questionnaire-based survey was designed by an MSc student who worked in the project also between 2020 and 22. So we used that questionnaire base to do the survey with slight modification. So a list of the nursery and plantation that has been surveyed was provided by TIPWG, and this includes nursery which produce forestry seedlings only, or in a combination with seedlings for other purposes like vegetables and ornamental. But however, only the data that are related to the forestry were considered to generate the result. So the forestry campaign who has plantation within the measured climatic region also has been surveyed. And the survey was conducted with several nursery and plantation which were willing to do the survey with us and participate in the program. So a total of 6 forestry nursery and 6 plantation campaigns took part of the nursery, and the information collected was related to 6 main categories for the nursery and 5 for the plantation. So according to each category, data collected were used to generate an updated comprehensive list related to the disease and the insect present in the forestry industry, I mean nursery and plantation, together with the list of pests and pathogens that are problematic, for which there is no available registration control measure or product. Additionally, for each of the pests and the pathogen, a reference list of control and management method used was also compiled, and an updated list of the pesticide used for their control in both nursery and plantation was generated for future use. So to date, the project has successfully fulfilled its first objective by accumulating an updated list of prevalent disease and insects that affect the forestry nursery and plantation, along with the chemical and alternative product deployed for their control. And all of those information has been available on the technical report that ICFR circulates among all of the collaborators. So next, please. So currently, as a progress, we also have a pre-screening trial at SAPI, at Clan Nursery, and we implementing powdery mildew and Campbellaria trial. And we testing different products, included fungicide, contact fungicide, systemic fungicide, organic systemic fungicide, natural fungicide. And the design we've used for all of those two trial is randomized complete block. And the number of the product we've been testing for powdery mildew, for example, is 6 different products, including all of those different categories that I mentioned earlier. And for Cambolaria, we are testing 15 products also that include all of those categories. And each of those trials has 3 replicates, and we're using nursery, SAPI nursery facilities, like I said. And for the data collection, we are looking for the disease frequency and severity. We are taking the canopy area and height. The insect inventory has also been taken. The phytotoxicity of each of the products we've been testing. The relative humidity and temperature also is a part of the data we are collecting currently. These 2 trials we started early January, and by end of this month, we should be finishing and produce the result to start with the following pre-screening still at SAPI nursery before we can go further to different nursery and different locations for registration purposes. Next slide, please. Okay, in terms of the output to date, in the nursery, we update the list of the insect and the disease, as well as their control measure. And through our survey, we collect the information that revealed that 8 disease and insect and 6 disease and 5 insect was found in the nursery and the field. And they've been counted as the most prevalent pests that affect both the nursery and the plantation. The list of the problematic pests for which no product has been currently registered for also has been collected. We also generate the list of the current control product, the biological control product, the natural and the synthetic product that has been used in the nursery and the plantation. And we also did the work by selecting the product from different functional groups for our pre-screening trial for Cambolaria at SAPI and powdery mildew as well. And like I said, we're doing currently the pre-screening trial for those alternative products we've been able to source. Please, okay. And for this year, this is a brief work plan I'm presenting to you because it's gonna be split with more details soon. So we have as an activity the pre-screening trial, which is going on at Klein Nursery for powdery mildew and Cambolaria. We aim to do a second pre-screening trial for the same disease, powdery mildew and Cambolaria, before the end of the season. And after that, we're going to continue developing and harmonizing the standardized protocol for nursery testing for those both disease. And later on during the year, we're going to do the pre-screening trial for the disease that is appeared in the nursery, which may be Botrytis cinerea or Rhizotonia, depending on what has been notified to us through all of our partners' nursery. And later during the year also, we aim to review to publish, a review paper and to do a publication. And there will be also an additional member to the team, which will be a MSc student to take some part of the research together with me. So that's what I can tell you for our work plan before we split it. I would like to thank everyone who who's still participating, in this project, and whoever is still being helpful to the team, and all of those one who has been supportive and guiding me so far. And I also want to thank TIPWG and FSA for founding, as well as SAPI, Mondi, FSA, Nelson Mandela, FABI, and the University of KwaZulu-Natal, with who I'm a postdoc. Thank you, everyone, and I'm here for the question. Thank you.
Thank you very much, Bernice, for your presentation. Really good work done so far. A huge amount of work also, and a very tricky project to manage, especially in terms of the seasonality of pests and diseases. And various such projects have been tried in the past, and as soon as you start a project, then the pest or pathogen disappears for a few months and things get delayed. So thank you for being willing to take on such a tricky project. I don't see any questions in the question and answer box for Bernice specifically, or in the webinar chat, but I always have questions. So Bernice, from your survey so far, were there any new pests or diseases that popped up in the survey since the previous survey?
No, Jolanda, I haven't seen new pest or disease that is not already mentioned from the previous survey. The only thing maybe that I can mention is that those disease or pests are not similar in every nursery. So some has those disease as a measure and the other one do not have it as a measure. Like Cambolaria, for example, only SAPI report that, Mondi do not have, and other nursery also do not have it. So there hasn't been new things from my knowledge.
That's great news. We have enough to deal with. Then I'd just like to, from my side, also encourage any of the other commercial companies, if you do have nursery space available for Bernice that she can use in the trials, especially when it gets to the next stage of Bernice's project. She'll need to include other nurseries, especially in other regions of the country also, in order to make her data usable by the pesticide companies. And then on that also, just to thank all the pesticide companies that have responded to Bernice's email so far, and especially those that have contributed with information and pesticides for testing in her first round of pre-screening. We really appreciate your collaboration there, and we look forward to communication with the other companies. And if any of the companies online have products that you'd like the industry to consider, please contact Bernice or Roger or Jackie or myself, and we'll get everybody together to look at that. Let me just double check, Bernice, if there isn't a question that's popped up.
There's one that's come through actually straight to me. I think they're having problems being online, and it's just to ask Bernice if her choice of target disease was based on survey results.
Okay, thank you, Kathy. Yes, the target disease has been based on the survey, because those survey, the result we got are from the managers. So the disease we've been targeting are the measure they have in their nursery. And that has been within all the nurseries. So we took what is the measure for all the commercial nursery and everyone involved.
And I could perhaps chip in from past discussions, also mentioned the seasonality of pests and diseases also. So that also, of course, plays a role as to what diseases have been looked at so far by Bernice. And as the seasons change, there'll be discussions. And then, of course, the big constraint here is this is a huge amount of work. So, of course, Bernice can't look at all the diseases at the same time, and we'll take it systematically. And hopefully in the future, like you said, you will be getting a master student joining, and hopefully then more diseases can be looked at. All right, Bernice, thank you very much for your presentation, and good luck with the rest of your work this year.
Thank you, Jolanda.
All right, I'm then going to hand over the floor to our next presenter, which is Jina Park from Stellenbosch University, who's going to tell us more about the project that she just recently started with regards to pesticides in forestry. So Jina, over to you.
Okay, good afternoon. I am Jina Park. My research topic is on weed detection and management in pine plantations using UAV-based remote sensing and machine learning techniques. Professor Bruce is my supervisor guiding this project, and my co-supervisor is Mr. Luca from Statistics Department, assisting with artificial intelligence. There are mainly 3 types of weed in the pine plantation. The known factors that limit the growth of the tree are high leaf-to-air vapor pressure difference and low water availability in the soil. Herbaceous broadleaf are the most concerning as they require the most water consumption from the soil. Currently, glyphosate applied with knapsack sprayer is the dominant weed control method in South Africa. Herbaceous broadleaf have the most significant effect in the competition of tree growth. However, if they are suppressed well during the first 2 years, weed management might be unnecessary from the 3rd year onwards. Due to the slower growth of the softwoods than hardwoods, weed management in pine plantation might be necessary until the 5th year. For these reasons, there is a need for the research on weed management in pine plantation. To obtain data, unmanned aerial vehicle can be utilized. They offer access to remote areas, higher resolution, real-time monitoring, and they are time and cost-effective. A few options are available for attaching sensors on UAV. Multispectral camera is suitable as it provides data with multibands. Machine learning is relatively new technology that is trained to identify relationships and patterns within given data, enabling it to make predictions. There are 3 types of machine learning: supervised, semi-supervised, and unsupervised. The unsupervised algorithm is the most complex and less accurate. However, once the model is built, it becomes time and cost efficient, especially for large data set like plantation images. This image is a supervised segmentation with labeling, where a box needs to be labeled manually for each seedling and weeds. In contrast, the right image is unsupervised with segmentation, where segments are automatically created based on the patterns found in the image. The problem statement of the research are as follows. Variable forest conditions compared to other crops, as tree plantations are widely distributed in South Africa. Delayed machine learning adaptation in forestry compared to other crop industries. For example, this image is the result of segmentation algorithm applied in cabbage identification. Also, there are challenges in implementing machine learning with large unlabeled data. The current weed control practices in South Africa are also inefficient as chemicals are manually sprayed all over the plantation, regardless of the weed density. The hypothesis of the research is that the developed machine learning model will enhance accuracy, efficiency, and adaptability in identifying weeds within tree plantation environments. The data will be collected using the UAV-mounted multispectral camera. Image augmentation is an editing of original image to train a model, which consists flipping, resizing, and changing colors of the original image. Grid creation and clustering is a method of organizing data with similar features into clusters. calculation of vegetation indices will be performed based on the requirements of algorithms. These are the recently available candidates of unsupervised segmentation algorithms. The image at the top shows the result of invariant information clustering, and the below shows the result of convolutional neural network. To build an unsupervised segmentation model, image augmentation is required to create features for every pixel. This may include color, intensity, and location. Then the model trains itself to identify pixels that are similar to each other. When similar pixels are grouped into a class, it is referred to be a cluster. After clustering, the patch with maximized mutual information becomes a segment. And this image is the result of image segmentation. To check the model's reliability, about 20% of the data will be manually labeled, then compared with the generated model's prediction. Mean intersection over union is employed to assess the quality of segmentation mask. A high IOU indicates good estimation. Boundary IOU compares the area within a certain distance from the segmentation boundary. The accuracy and F1 score will be presented in percentage, calculated using confusion metrics. The expected scientific contribution of this research are as follows. Real-time monitoring of weed growth, as it has already been applied in other crop fields. Tracking herbicide uses by calculating weed density. This will result in improving weed management and saving management costs. Automation of work process, including weed detection and chemical spray, will be applied. Lastly, conducting this research will contribute to the development of precision agriculture techniques in forestry industry. Thank you for your time and attention. Many thanks to the sponsors, FSIF and FSA, and the research working group, TIPWG.
Thank you very much, Jina, for your presentation. I'm just having a quick look to see if there's any questions. Don't see anything in the question and answer box or the webinar chat. Please get them in if you have any. From my side, just you mentioned data, and that getting enough data is always a challenge. For your project, Jina, in terms of data, you know, what number of images are you looking at, and that you'd say would be sufficient data to build an accurate model?
I am thinking about at least 3 department plantations to get a variable to train the model. So each plantation might be 5 to 10 hectares, and I will get UAV images.
All right, thank you. So yeah, and then from each compartment, obviously you need to have multiple images. So how many images? I know for some of these AI projects, they talk about thousands of images required to get sufficient data.
Yes, normally for one compartment, it is required about 600 images, but after getting the data, I merge all the images to make one image. But yeah, I would say from 500 to 700 per compartment.
Thank you, Jina. Let me check. No other questions from anybody. There's just a comment from Oliver Wood that says, thank you for your exciting presentation, Jina, and nice to meet you.
Thank you.
There we go. Thank you, Jina. Then thank you also to all 3 presenters again for taking the time to prepare presentations. It's always a daunting task. Then here we go. Here's another question that just popped in for you, Jina, from Marius ter Blanche, asking whether different types of weeds require different AI models.
No, it will require only one model to differentiate different types of weed, because I will train all different ways to one model, and the one model can define, distinguish the different ways.
Thank you, Jina. Yeah, so last word from me. Thank you to everybody. And as you heard from these three presenters, many different organizations and people involved in this work. And as an industry, we appreciate every single person that's involved and every single organization. So big word of thank you, especially to our pesticide companies also. We cannot do this type of work without you. And we really appreciate the support. And then also to all the research institutions, thank you so much for putting time and effort and resources and students into projects like this. It's greatly appreciated. So from me, over to Katie. Thank you.
And also, Jolanda, a massive thank you to you, not only for hosting this segment, but for what you do for TIPWG. Obviously, Jolanda's role in TIPWG is a voluntary one. She does our research coordination, and we very much appreciate her and the time that she spends doing this aspect of the TIPWG work. So thank you, Jolanda. Just quickly, before we move on to the next segment, could all of the people who've presented their research just quickly